Hello everybody, this is Viji. Welcome back to Easy Chapters. Hope you all are going great. Today I am with a new topic, that is a topic from science. Today we are going to discuss a topic from 8th grade, that is cell structure and functions. Let us see the topic. Let's quickly see the objectives of today's study. First one, to study the cell and its variations, to understand structure of cell and the nucleus, and finally to compare the plant cell with animal cell. When we look around, we can see so many types of constructions, right? They will be differing in their design, their shape, size and everything. But there is a basic material behind that. There is a basic building material for all these constructions. What is it? It's a brick, right? So a brick is a basic building material or a basic structural material for any constructions, right? Like that, all the organisms are also having such a basic structural material, and that structural material is known as a cell. So we discussed bricks are the basic building materials of any construction. Like that, a cell is a basic structural and functional unit of any organism. As how a brick contributes in the formation of a building, like that only a cell contributes in the formation of an organism. So in short, we can say that cell is a basic structural and functional unit of an organism. The cell was first time discovered by the scientist Robert Hooke with a very simple magnifying device, and the material was a cork. He observed a piece of cork, a slice of cork under his magnifying device. He found that there were honeycomb-like structures seen, and he named these honeycomb-like structures as cells. So we studied about cell as a structural and functional unit of any organism. Every organism is made up of the basic structural unit cell, and it was invented by Robert Hooke in 1665. Now my question is, how do a cell look like? Is that all the cells are alike, or is there any difference between different cells that we have to study? Actually, cell exist in different forms. It may vary in their size, in their shape, and also in their number, that we will see in detail. There are different types of cells based on their number, shapes and size. We can see each one. First of all, we will see how the cell varies according to their number. Based on the number, organisms are categorized into two forms. They are unicellular organisms and multicellular organisms. So the first type is unicellular. Unicellular organism. Here from the name itself we can understand what it is. Uni means single. Cellular means cells. So organisms that are having only one cell in their body. The entire body is having only a single cell. That is known as unicellular organisms. And here example is amoeba. Amoeba is an example. Paramecium is an example. That is known as unicellular organisms. And here the speciality is that the organism is having only a single cell in their body, and that single cell is enough or capable to carry out all the activities of that organism. Respiration, excretion, digestion, everything. It is possible by that single cell only. That is known as unicellular organisms. That is organism that is having only a single cell in their body. This is a picture of amoeba and paramecium, the unicellular organisms. Next type is multicellular organisms. Here also we can see that the term multi means many, cellular again it is cells. Multicellular means many cells. So organisms that are having many cells in their body are known as multicellular organisms. Any higher organism, any complex organisms are coming under multicellular organisms, like human beings. So human beings are an example for multicellular organisms. Even in multicellular organisms, they are having so many cells, multiple number of cells are there, but in the initial stage there will be only a single cell present. First of all, there will be a single cell, and that cell will be undergoing repeated division. That cell will undergo repeated divisions to form a number of cells or a mass of cells. and such cells, similar cells, will be grouped together or assembled together to carry out specific functions in the case of multicellular organisms. But in the case of unicellular organisms, there will be only a single cell, and that cell is carrying out the activities of the organism. The organism will be very lower form, so that the activities also will be in the lower grade only. So that single cell is enough to carry out all the activities of that organism. Whereas in the case of multicellular organisms, the organism is bit more complex. The activities also will be more complex. Here the cells will be clubbed or assembled together, and it will be carrying out diverse functions. Now we will see the types of cells based on their shapes. There are various shapes for cell, and we will see them. The first one is an irregular shaped cell. This is an irregular shaped cell. This is seen in the case of amoeba. Amoeba is an organism, unicellular organism, and it doesn't have a proper shape. The shape will be changing every now and then. That is known as irregular shape, and this type of cell is seen in the case of amoeba. Amoeba is an example that is having irregular shaped cell. This is a round cell. Round cell is seen in the case of female reproductive cell, that is ova, otherwise known as egg. So egg is a female reproductive cell that is round in shape. Now this is a male reproductive cell, otherwise known as sperm cell. Sperm cell, and here you can see the shape. It is having the shape of a tadpole. Tadpole shaped or tailed cell. Our next cell is an elongated one. It is having branches as well, right? So this is an example of nerve cell. Nerve cell, it is branched and also elongated. Next one is spindle shaped cell. Spindle shape means the ends will be thin and it will be tapering towards the end, and in the middle portion it will be broad. So such a structure shape is known as spindle shape, and this is seen in the case of muscle cells. So spindle shaped cells are seen in the case of muscle cells. Now comes the column cells, columnar cells, that is arranged in the pattern of columns. These cells are seen in skin cells. These are skin cells. This pattern of cells are seen in the case of skin. So skin cells, they are having a columnar pattern. Then again it is a spherical cell. Spherical cells are seen in the case of blood cells. Blood cell is an example of spherical cell. So there are various shapes for cells. It may be irregular, then round, tadpole shape, then elongated branch type, spindle shaped, column type, or even spherical cells are seen. These are the types of cells based on their shapes. So we studied that cells can exist in different shapes, and it is because of the presence of the outer membrane. This outer membrane is giving that peculiar shape to the cells. So let's move on to the next topic, that is the varieties of cells based on their size. What will be the size of a cell? Any idea? A cell can be very minute, right? A cell can be very minute and its measurement may be some micrometers, a few micrometers. And what is a micrometer? It is just 1/1,000,000th of a meter, right? So that much minute a cell can be. Now there are few large cells also. Large cells can be measured in centimeters. So the range is between micrometers, few micrometers to few centimeters. In between comes the size of a cell. Now if the cell is very small or minute, we cannot see it with our naked eyes, right? Then how can we see that? We need microscopes. And if it is bit large, then we can see these cells with our eyes. So a cell can be very minute and about few centimeters. So a bacterial cell is the smallest cell. It may be measuring around some 0.1 to 0.5 micrometers only. So bacterial cell is the smallest cell. Now which one will be the largest cell? The largest cell is the egg of ostrich. Egg of ostrich is having a measurement of around 170 into 130 millimeter. So egg of ostrich is considered as the largest cell. Not only egg of ostrich, other eggs like hen's egg, all these eggs are visible with naked eyes. We don't need any microscope to observe such eggs. For minute cells, definitely we need a microscope. So we studied about the types of cells based on number, shape and size. Now my question is there any relation between the size of a cell and size of an organism? Like for higher organisms, the size of the cell will be larger, and for smaller organisms, the size will be smaller. Something like that. Is there any relation is there? No. There is no such relation between the size of the cell to the size of an organism. But there is a relation between size of the cell to the function of the cell that we will see. Let us take an example, nerve cell. This is a nerve cell. It is highly branched and also elongated cell. Why? Because the function of nerve cell is control and coordination of the body. It is to transfer messages from brain to all parts of the body and vice versa. So, for that purpose, the nerve cell is long and branched. So, the size of the cell is related to the function of the cell. Now, let's see another example. This is a spindle cell. Which one it is? Muscle cell. Muscle cell is having a spindle shape. And what is the speciality of spindle shape? It is tapering towards both ends and the middle portion is broad. Now, what is the function of a muscle cell? Muscles mainly it is for contraction and relaxation, and the same function is carried out by this cell. The shape indicates that it can be compressed and relaxed. Such a shape it is having, that is a spindle shape. Now comes skin cell, that is existing in the form of columns. Columnar cells are seen in skin. And what is the function of skin cell? The main function of skin is to cover the body, to protect the body. And here the cells are arranged in a form of barrier, right? It is arranged in the form of a barrier or sheet. So here also the size of the cell is related to the function of the cell. Understood? So we studied about cell. Cell is a structural and functional, basic structural and functional unit of an organism. Now, from a cell, how such a complex organism is developing? That is a question, right? For that, let's start from cell. What is a cell? It is a basic structural unit of an organism. Now, these cells, after undergoing repeated divisions, a mass of cells are formed. And such a mass of cells that are having some common properties, maybe having common functions, common properties, such cells will be assembled together and it will be giving rise to a new level, that is known as the tissue. It will be developing into a new structure, that is known as the tissue. So from the groups of cell or the assembling of cells, a new structure is forming, a new level of development is formed, that is known as the tissue. Now what happens to the tissue? Again, the same process is happening. It has to cover the next stage. It has to go through the further development stages. So tissues, there also will be similar tissues are there. They will be assembled. Similar tissues will be assembled and they will be giving rise to an organ. So organs are developed from tissue. So starting from the cells, cells assembled to form tissue, tissues assembled to form organ. Now what happens? Similar organs are there. Similar cells will be giving rise to similar tissues. Similar tissues will be giving rise to other organs also, right? So there will be other organs also will be formed. Now organs that are having similar functions, they will be grouped to form an organ system. So from the organs, the next stage of development is organ system. Now after the organ system, what will happen? There will be several organ systems are developed, and after the system development, the entire organism is finally produced. That is organ systems. All the organ systems will be contributing the formation of an organism. So starting from the cells, mass of cells are formed. These cells will be assembled and it will be giving rise to the next stage of development, that is tissues. Similar way, tissues also will be assembled based on their properties and function. It will be giving rise to an organ. Such similar organs will come together. It will form an organ system, and organ systems, there are various systems of organs are there in our body, and all the organ systems finally contribute in the formation of an entire organism or the body of an organism. Let me explain with an example. Nerve cell. Nerve cell is a basic cell. So nerve cells, it will be undergoing so many divisions and mass of cells will be formed. Now these cells will be assembled and it will be going to the next stage of development, that is a formation of tissue. From the nerve cell, which tissue will be developing? It is the nervous tissue. So from nerve cells, nervous tissue is developing. Now tissue will be further assembled and it will be assembled to form the next level of development, that is known as the formation of organ. So from nervous tissue, which will be the organ developing? It will be brain, sometimes spinal cord, right? These are the organs that can be developed from a nervous tissue. So organs are developed. These organs that are having a common function will be grouped and it will form a system. What is a system? That is formed with this brain, spinal cord, nerves and all. That is the nervous system, right? So here the system is developing, right? Like that, we started from a nerve cell only. Like that, several other cells are there, right? Skin cells are there, muscle cells are there, so many cells are there. All these cells will be passing through the same stages of development and they will be contributing to corresponding organ systems. And once all the organ systems are developed, the entire body of an organism is completed. From a cell, after undergoing various stages of development, an organism is developing. Now we will study the structure of a cell. What are the different components of a cell? There are three major components in a cell. They are plasma membrane, cytoplasm and nucleus. Let us see through a diagram. A cell is a very simple structure. This is a diagram of a cell, a very simple structure. So cell, it is having an outer boundary that is known as a plasma membrane. This one is the outer boundary, the limiting membrane, that is known as the plasma membrane. It is otherwise known as the cell membrane. Now towards the inner side of the plasma membrane, inside the cell, there is jelly-like material seen, that is known as the cytoplasm. This jelly-like fluid or material that is seen inside the cell is known as a cytoplasm. Now there is a dense round body seen in the middle of the cell, that is known as the nucleus. So there are three major components in a cell, that is the outer boundary, that is known as a plasma membrane or cell membrane. Inside the cell there is a jelly-like material seen, that is known as a cytoplasm, and in the center there is a dense round body seen, that is the nucleus. Now the plasma membrane is little bit porous in nature so that materials can enter into the cell and leave the cell. So these are the three major components seen in a cell. In addition to that, there are some organelles seen that will be studied in later classes. So we discussed about cell and the structure of cell. So we told that the cell is having three major components, that is the plasma membrane, cytoplasm and nucleus. Now, is it possible to see a cell and these minute structures? Is it possible for us to see? That is a question, right? It is possible, but for that we need some equipments. Let us see what are the different equipments that we need to see a cell in detail or to study the structure of cell in detail. So in order to study the detailed structure of a cell, we need few things. First thing is microscope. Then second we need stains or dyes, and the third one is slides. Now we'll see what is a microscope. What is a microscope? Microscope is a lighting device. It is an optical device that is used to see a very small object in a magnified way. Magnification is a principle behind microscopes, and how it is possible we will explain with the diagram. Then comes the stains and dyes. Stains or dyes. Because these materials, these cells, it will be colorless in nature. So we need to color them to see and differentiate the inner components of the cell. For that purpose we are using stains. Then comes the slide. That is a rectangular glass sheet on which we are keeping our specimen and we are viewing the material. So these are the three things that we need to study the structure of a cell in detail. That is microscopes, stains and slides. These are the three necessities that we need to study a cell structure in detail. microscope, dyes, otherwise known as stains, and slide. First of all, we will see what is a microscope. Though this portion is not included in the syllabus, I feel better to explain this. Microscope is a laboratory device that we use to see small things in a magnified way. Let us see the parts of a microscope. First of all, there is a body tube, and on the upper side of the body tube, there is a lens known as eyepiece. Otherwise, it is known as ocular lens. And on the underside of the body tube also, we can see lenses that are known as the objective lens, and it has got the powers of 10x, 40x, 100x like that. The power of the eyepiece, that is a topmost lens, is also 10x. Now we can see two adjustments attached to the body tube. The large knob is known as the coarse adjustment, and the small one, just beneath the large one, there is a small knob that is known as the fine adjustment. Now coming to the lower part, there is a mirror is there. Then on the upper part of the mirror, there is a platform is there with clips, and in the center of the platform we can see a small glass aperture is there, an aperture that is covered with glass. Now this is a platform on which we are keeping the specimen to be studied. Now there is a mirror. Now we will see how a microscope works. First of all, we need a light place. We need a room with plenty of light to keep the microscope. We have to keep microscope in a place where we are getting enough light. Keeping there, we need to adjust the mirror so that light is reflecting on touching the mirror. It is reflecting to the platform. Now in the center of the platform, there is an aperture with glass, and above that only we are keeping the specimen. Now after reflecting from the mirror, the light is passing through the specimen or the substance we have kept here to study. Now the light will be moving on to the objective lens. So the objective lens will be capturing the light that is passing through the specimen, and the first image is formed here. After passing through the objective lens, first image is formed. Now the image, the first formed image is passing through the body tube. Light from the first image is passing through this body tube, and again it is reaching to the second lens, that is the eyepiece. Now a second time magnification is taking place. So two times image is getting magnified. First time it is getting magnified by the objective lens, and finally by the eyepiece. So a small object is getting magnified twice, and the final image is produced by the eyepiece. Now we will study about the magnification produced by the microscope. For that we need to understand few more things. That is, objective lens are having different powers, that is 10x, 40x, 100x like that. So if we want to view a thing in low power, we need only 10x power. But if we want to study further details, we have to go for the high power, that is 40x, 100x like that. 100x are all not available in the normal laboratory microscopes. That is seen in a highly sophisticated microscopes only. So objective, the power of objectives are 10x and 40x normally, and the power of eyepiece is normally 10x. Now how to find out the magnification? So magnification is a product of the magnification produced by the objective lens and the ocular lens. So if we are choosing a 10x of the objective, then magnification will be this 10x into the 10x of eyepiece. So total magnification will be 100x. Now if you are using the 40x of the objective, now the total magnification will be 40x of objective lens into 10x of the eyepiece. Now if it is 100x of the objective, total magnification will be 100x into 10x of the objective. Thus two times the magnification is produced, and the final magnification will be the product of the magnification produced by both lenses. So that much will be the extent of magnification produced by a microscope. In short, we can say that we are able to see a minute thing in many times more than its real size. So moving on to the next component, that is dyes, otherwise known as stains. Dyes or stains are also used for cell study. Why? It is because the cell is mostly transparent or colorless in nature, then how we will differentiate the cytoplasm, nucleus, etc. For that purpose, we are using the stains. And by adding the stain, the cell will take up the color and it will be easy for the study. Now there are two types of stains. One is nuclear stain and the other is cytoplasmic stain. Nuclear stains means these stains will be coloring the nucleus only. If we add the stain to the cell section, nucleus only will be getting stained. Next is cytoplasmic stain, that will be giving color to the cytoplasm only. Now there are few examples like methylene blue, safranin, all these are dyes that are commonly used in laboratories for cell studies. Then comes the third thing, that is the slide. It is nothing, just a rectangular glass sheet, glass piece it is, actually a glass piece it is, on which we are keeping our specimen, whether it is an onion peel, cheek cell, anything, we have to keep it on the rectangular slide. Then after that we used to keep a small thin film of glass over that to keep the material intact in position, and after that only we are keeping the material on this platform. So these are the three things that we need to study a cell in detail. Once our equipments are ready, then what we need? We need a material to study the cell structure. For that, I have a carrot with me. What is the speciality of carrot? Carrot is a root, right? It's a root modified vegetable. So to study the structure of a carrot root, what we need? We need a thin section of the carrot. For that, I have already cut one thin section. This is a section, this is a cross section of the carrot root. Now what we have to do? We have to keep this material or a portion of this material in the slide. Then we need to stain it and we have to keep it in the stage of a microscope and we have to observe. Then we can see the fine details of the root cells. When we observe this thin slice of carrot under a microscope, we can see that inside this carrot piece there will be arrangements of cells in this way. Along with the cells there will be elements like xylem, phloem, etc. All those things will be there, but mainly the arrangement of cells we can see inside the slice will be like this. It will be tightly packed cells will be seen in the microscope. That is in a low magnification, but when we are going for a higher magnification, we can see that each of the cell will be having a boundary like this and there will be center nucleus will be there. So this is how a cell appears in the microscope. Another material to study the structure of cell is an onion slice. From this onion slice we can take the peel of onion. We can take the peel from the inner portion of the slice. It will come like a transparent sheet. Hope you can see this. This is an onion peel and we can cut this peel in a perfect shape, and after that we can stain it. And we can observe this under a microscope. In that case also we can see the cells, tightly packed cells, both in low power, that is in a low magnification and high magnification. We can see the detailed structure of a cell. Like that, another example is the human cheek cells. Human cheek cells means we can scrape the epithelial cells inside our cheek with a toothpick. The same way we can keep it in a slide, then after that we have to stain it and we can observe it under the microscope. Then we can see how the human cheek cells are looking like. This is how the onion cells and cheek cells appears after staining. When we study nucleus in detail, we have to study few more things. That is, the nucleus that is in the center of a cell, it is having an outer membrane. The nucleus is covered with an additional membrane that is separating nucleus from the cytoplasm. That membrane that covers or that lines the nucleus is known as nuclear membrane. So this is a nuclear membrane, and this nuclear membrane is also porous in nature. It will allow the transport of materials from and to the nucleus. So the outer membrane of the nucleus is known as a nuclear membrane. Now what all things are seen inside the nucleus? We can see a round body cell, a large round structure is seen inside the nucleus, right? That is known as nucleolus. So The large round body that is seen inside the nucleus is known as a nucleolus, and its function is regarding the protein synthesis, and that we don't have to study now. At present we just want to know there is a round structure inside the nucleus and its name is nucleolus. Now what is left there? We can see strands, thread-like strands are seen, and they are known as the chromosomes. And what are chromosomes? It is in the chromosomes the units of inheritance, that is genes, are located. And what is a gene? Sometimes you used to say, I have got resemblance like my mother, I have got curly hair like my mother, I have got black eyes like my father. How it is happening? We are getting or we are inheriting the traits of our parents. It is through the genes. We are inheriting genes from our parents. That's why we are getting resemblances like our parents. So these genes, being the units of inheritance, these genes are located in these chromosomes, and this is the detailed structure of a nucleus. This is a structure of nucleus. Now in the center we can see blue colored strand like structures. They are the chromosomes. Chromosomes are actually thread like structures on which the units of heredity, that is genes, are located. Now what is a gene? Gene is actually the basic unit of heredity that decides what all traits we should get from our parents, in what all traits we should resemble our parents, what all characters we should get from our father, our mother. All these things are decided by the genes. So genes are the units of heredity, and that is coded in the chromosomes. Now genes forms a part of the DNA. DNA is the genetic material. Now based on the presence of the nuclear membrane, organisms are categorized into two groups. They are prokaryotes and eukaryotes. Prokaryotes, the word can be splitted. Pro means primitive and karyote means nucleus. So prokaryote means those organisms that are having a primitive nucleus. They are known as the prokaryotes, and examples are bacteria, blue-green algae, etc. Now comes the eukaryote. Eu means true. So eukaryote means true nucleus. Organisms that are having a true nucleus, real nucleus. They are known as the eukaryotes, and all the other organisms except bacteria and blue-green algae are eukaryotes. So in prokaryotes we can see that if it is a cell, the genetic material will be freely floating like this. There won't be a nuclear membrane. But in the case of a eukaryote, we can see that there would be a distinct nucleus with nuclear membrane. So based on the presence of nuclear membrane, organisms can be classified into two types, prokaryotes and eukaryotes. If the organism is not having a prominent nuclear membrane, the nuclear membrane is absent, they are known as a prokaryote, and if there is a prominent nuclear membrane present, they are known as the eukaryotes. This diagram shows the eukaryotes and prokaryotes. As we discussed, we can see that in prokaryotes there is no nuclear membrane in the middle. So here we can see that the nuclear material is simply scattered in the middle. The main living things are plants and animals, right? So is there any difference between the plant cells and animal cells? Cells are present both in animals and plants. Is there any difference between the cells of plants and animals that we have to study? So let us go for a comparative study of the plant cells and animal cells. Moving on to the last topic of this unit, that is comparison between plant cells and animal cells. Let us see whether there is any difference in the cells of plants and animals. Let us see. So these are the two structures, two diagrams. One is plant cell and another is animal cell. Now, which are the basic components of a cell? What are the basic structures of a cell? We know they are the cell membrane, cytoplasm and nucleus. Let us start with the plant cells. This is a plant cell diagram, and here we can see the outer membrane. Here we can see there are two membranes. Limiting membranes are two. So which are they? The outermost layer is the cell membrane, right? No, but in the case of plant cells there is a difference. The outermost membrane is known as the cell wall. Now the second layer, towards the inner portion of the cell wall there is a membrane. That is a cell membrane. So in plants, the outermost layer is the cell wall, and towards the inner portion of the cell wall there is another membrane, that is the cell membrane. The rest of the things are same. Jelly like material is the cytoplasm. Now there is the nucleus, round shaped dense structure. Nucleus is there. Now here we can see a hollow space, that is a vacuole. That is a vacuole is there. Now we can see colored objects in the cell. What are they? They are known as the plastids. Colored substances are known as the plastids. They are various in colors. Now few of them are green in color. So green in color means that plastids contain chlorophyll, and those plastids that contain chlorophyll are known as chloroplast. So there are two membranes outside. The outermost layer is known as cell wall, not the cell membrane, it is the cell wall. Now towards the inner side of the cell wall there is another membrane, that is the cell membrane. Then jelly like material, that is the cytoplasm is there. Nucleus in the center, dense round body, nucleus is there. Then we can see a large vacant space is there, that is known as the vacuole. Then towards the inner portion of the cell we can see colored pigment like structures, colored bodies are there. They are known as the plastids, and few of them are green in color, and green colored plastids means they contain the pigment chlorophyll. Why? Plant cells have the function of photosynthesis, and for photosynthesis they need chlorophyll, right? So plastids that store chlorophyll are known as chloroplast. These are the structures of plant cell. Now coming to the animal cells, here outermost membrane is there, that is the cell membrane. There is no double layer, so cell wall is absent. There is cell membrane, that is the outermost layer. Now there is the jelly like material, that is known as the cytoplasm. Now towards center we can see the dense body, round body, that is the nucleus. Then we can see small spaces are there, they are the vacuole. Vacuole is there. Then in addition to that there is no more structures in comparison to the plant cell. Now what is the difference here? So these are the things there. We can see that there is a cell membrane, there is cytoplasm and nucleus in common. Then difference is that in plant cells we can see that there is a large vacuole is there, but in the case of animal cells the vacuole is small. Then chloroplast. Chloroplast is seen in the case of plant cells, but in the case of animal cells there is no chloroplast because there is no photosynthesis. Now cell wall. Cell wall is the limiting layer in the plant cells that is giving a perfect shape and rigidity to the cell. Whereas in the case of animal cells, the cells are not having that much a particular shape or a solid shape. Irregular shapes are also seen in the case of animals. So there we cannot see any cell wall. The outer membrane is cell membrane. So in comparison we can see that the difference is regarding the cell wall, chloroplast, and the size of the vacuole in comparison with plant cells and animal cells. Rest, the presence of cell membrane, cytoplasm, nucleus, all these things are common in both. So that's all about this topic, cell structure and function. Hope you all understood the topic. If you like the video, please share and subscribe the channel. I'll be back with another video. Till then, you all take care, stay safe. Bye.